This conference will now be recorded.
All right. Well, welcome everyone, and welcome along to this is the first of the webinar series we are initiating for the Sheep Reproduction Strategic Partnership. And our speaker today will be Will van Wettere from the University of Adelaide. But before we start with Will's talk, I thought I'd just give you a bit of an overview of what the Sheep Reproduction Strategic Partnership is all about, and so you understand a bit more the purpose of the webinars and that we're starting today. But just a bit of housekeeping to start with. So thanks very much for joining. Please make sure, as you can see, I can see you're all muted, which is great, and your cameras are off. If you're having any trouble with audio, just try and call in that number that's listed there. And if you want to ask a question, most we're all pretty familiar with using online meetings and things these days, but just use that chat function. So just have a look, you can see, you can click that little chat bubble up the top there, write your message down the bottom, and then don't forget to click send. And then I'll keep an eye on the chat function. I'll direct all those questions to Will at the end of the webinar. So the Sheep Reproduction Strategic Partnership, the goal of the partnership is to profitably and sustainably increase lamb production through increasing weaning rates and decreasing mortality. So we'll do this by building on existing research, development, adoption. We're going to focus on and promote on-farm best practice management and work with producers to apply R&D on-farm in a way that's practical, economical, and that works for different business models. We'll be testing and demonstrating supporting technologies and identifying and filling gaps in R&D. And as you can see from the diagram there, we'll be working right across the entire reproductive cycle to improve reproductive performance and provide solutions for producers. So the goal of the partnership is to foster collaboration across the sheep industry. We want to start to develop larger long-term projects that address a common goal and deliver benefit and impact to the sheep industry. To start with, we now have a website that's live as a page on the MLA website, and at the moment it just gives you the basics about the partnership and the key contacts. But over time, that will become our one-stop shop for sheep reproduction information, and you'll find more and more information on there as time goes by. As I said, this is the first of our webinars for the program, and so the recording of today's webinar will be made available on the website later on. And also, you may have seen the sheep reproduction RD and alerts that I put out on a monthly basis. It just captures the latest in sheep reproduction related research and then sends those out to advisors, producers and others in the industry to try and foster that collaboration and get a greater understanding of the research that's been undertaken in Australia and around the world and put people in touch with each other. And actually today's webinar really came out of the feedback I got from the alert back in February when I included a couple of papers from Will's team at the University of Adelaide that they'd published on supplementing merino ewes with melatonin and the impact on twin lamb survival. So today's webinar was really a function of that feedback I received from that alert that was back in February. So as I said, today's webinar is going to be presented by Will van Wettere, and I'll just make Will the presenter. So that, and while I'll just find him on my list, where is he gone? There he is. So while we're doing the switchover, I can introduce you to Will. He's a senior lecturer at the School of Animal and Veterinary Science at the University of Adelaide. And when he's there, he works on developing management strategies to improve the reproductive performance of female production animals, sheep, pigs and cattle. He completed his undergrad degree at the University of Edinburgh and then worked on dairy farms in New Zealand and South Australia. He completed his PhD at Adelaide University on the reproductive physiology of sows and then undertook a 3-year postdoc position with the Pork CRC. And he was appointed to his position at Adelaide Uni in 2009 as a lecturer. So I'll hand over to you now, Will. So it's all yours.
Okay, thank you. So can you hear me okay? Can you hear me okay? Everything all right?
Yes, I can.
Yeah, excellent. And my slides are up? Brilliant. Okay, so before I start, I'd like to thank soon the SRSP and MLA for the opportunity to present what I am going to present today. And I'd also just like to acknowledge the funding bodies, so MLA Donor Company, Sheep Industry Fund, SARDI, PIRSA, and the University of Adelaide, in particular Davies Livestock Research Centre, as well as my colleagues who have done a lot of this work with me. So people like Jamie Daly, Tom Flynn, Elise Swinburne, Karen Kine, Alice Weaver, Jen Kelly, and the evergreen Dave Kleeman. So now I'll probably just get started. So the focus of today's talk, I'm going to talk a little bit about the work we are doing to ensure that lambs like the one on the left, with all the meconium staining on him, he's clearly had a difficult transition into life. It's been a hard parturition process, and I'm going to talk about the work we are doing to try and make sure this lamb survives and ends up like the one on the right, which is a more viable lamb and it has a much higher chance of surviving. So the problem we're trying to fix, and apologies if this is teaching some of you to suck eggs a little bit, is that too many lambs die, basically. High lamb mortality rates represent a significant risk to the Australian sheep flock. I think recent estimates are around about $540 million it costs in lost production. And in addition to this economic impact, it represents a fairly major welfare issue. And the reason why the cost is so high is that about somewhere between 10 and 40% of lambs will die between the start of the birth process and marking time or weaning time. And if you break that down into singletons and twin lambs, we'd expect something between 13 and 21% of singleton lambs to fail to survive and around 21 to 38% of twin lambs to fail to survive. So we then start to hone down on when and why lambs die. We know from the literature, from surveys, that around 70% of lambs will die within the first 3 days after birth. And of these deaths, most of them can be attributed to dystocia, so birthing difficulties, and then starvation, mismothering, and exposure, so the SME complex. And if you look at that across the different breed types, a recent survey from New South Wales suggests around 70% of deaths are attributed to these causes in merino ewes, about 59% in terminal ewes, and around 67% of crossbred ewes. So then if we look down at just in a little bit more detail, is why do lambs die? Well, if you just take a minute to look at the pie chart on the right-hand side, we can see that birth-related issues are responsible for around about 50% of deaths. Starvation, mismothering is probably around 25% roughly. In utero, so before birth, premature births, deaths in utero, around about 10, 15, 12%. And then we have things like predation, a small fraction, cold exposure, and other causes. And what we can kind of deduce from all of this is that the birth process does significantly impact the ability of lambs to survive. So it affects their viability, their vigor, how they interact with their mum. And the difficult births will not only end up with due to stillborn animals, so dead lambs, they'll also end up in delayed deaths and increase their chances of dying due to starvation, mismothering, and exposure. And what we kind of would learn to understand is that the birth process is inherently dangerous for lambs, any neonate really. All lambs, like other neonates, they'll experience some degree of oxygen deprivation or hypoxia during the birth process. And this degree of this hypoxia increases with labor length, so the longer it takes from the start of contractions through to when they're expelled. And the degree of hypoxia by inference is going to be higher in multiple births. The consequences of these hypoxia are really quite severe. It ends up in things like damage to the brain, so brain damage to the nervous system, other vital organs can be damaged, kidneys, etc. You end up animals with impaired neuromotor activity, impaired ability to seek the udder, so impaired udder-seeking behavior. They're probably less vocal. They take longer to stand and suckle, and therefore they're less likely to consume colostrum or enough colostrum in the required time frame, so within about 12 to 24 hours of birth. Hypoxia impairs the thermoregulatory ability of the lamb, and all of these things also compound together to increase the chance of the ewe leaving the lamb or one of the lambs that are born if there's two of them. And that basically, sorry to sound like a harbinger of doom, but it all ends up increasing the likelihood of death of the lamb. So if we just think about the cost of birth hypoxia, and these are kind of back of the envelope kind of maths, if we accept that roughly 70% of early lamb deaths can either be attributed directly or indirectly to birth hypoxia, and based on a mob of say 1,000 ewes with a 30% twinning rate, we may expect to see something like 115 to 200 dead lambs as a result of birth hypoxia. So that kind of sums up the problem. And now I'm just going to talk about the objectives of the work we're doing at the moment. In about 2018, we secured a project, luckily to secure a project through MLA, entitled Novel Strategies to Increase the Weaning Rate of the National Flock, and it's a collaborative project between the Davies Livestock Research Centre and SARDI. And the objective of this project is basically to increase the ability of twin lambs to cope with birth hypoxia. So we're aiming to try and increase the vigour of lambs, make the lambs more vital, and increase their ability to thrive when the environment is challenging. And hopefully that will result in more twin lambs being weaned. And the way we wanted to achieve this, or are hoping to achieve this, is by supplementing twin-bearing lambs during the second half of pregnancy. We've focused on 4 compounds: caffeine, rumen-protected amino acids, betaine, and melatonin. And melatonin is the focus of today's talk, but I will talk you through the basic premise of the trial, which applies to melatonin equally as to the others. So for each of the compounds, in this instance melatonin, there are 3 stages to the project. There's a stage 1, which is basically where we do intensive research. Animals are managed outside till about 30 days before they're due to give birth. They then come into an animal house where they stay in individual pens, they lamb inside, they're under constant video surveillance, and this gives us the chance to record a whole thing, a plethora of data related to indices of lamb survival, including lamb survival itself. So things like time to stand, time to suckle. We can measure colostrum intake in the lamb based on IgG levels in the blood. We can measure colostrum, we can get colostrum samples from the ewe, and we can actually measure the course of parturition as well. Compounds, in this case melatonin, which show positive effects on lamb survival or indices of lamb survival, go to what we call stage 2, which is where research is taken under in field research settings. So SARDI research sites, so Minnepa in this case, and other stuff is being done at Kaibi, Baliti, and Struan. And under these trials, ewes will lamb outside, and the number of measures we take will much reduced, and they usually go down to things like birth weight, timing of birth, and more detailed measures of lamb survival, so we know how many lambs are born, when they die, and how they die. And compounds which successfully pass that stop-go point, which show improvements in lamb survival under field research settings, will go on to commercial validation. So they'll basically be tested within commercial partners, where all we'll do is just look at marking rates primarily, and maybe some other measures depending on what is possible within that system. So that brings us back to why melatonin, why we're interested in it. Melatonin is a hormone secreted by the pineal gland in the brain. It's secreted at nighttime, and it's well known for regulating circadian and seasonal rhythms. Some of you may use it or have used it in time ways to bring on the breeding season early in ewes, for example. It also, in addition to that, we're finding out it has a lot of other roles within the body and in lots of animals. It's even in plants. It's present in plants as well, but we believe it has neuroprotective properties. It works as a potent antioxidant to minimize the impacts of hypoxia in the lamb brain. We know that from studies focused on human research, where they have challenged the lamb quite severely and found that melatonin prior to and after the challenge has a very positive effect on the lamb's growth and the brain's ability to cope. The benefit of it is that it does freely cross the placenta, which means that we can give it directly to the fetus via the mum, and we don't have to give it to the fetus directly after birth. Like I said, previous studies have been focused primarily in research focused towards improving pregnancy outcomes in women, and they found that supplementing pregnant ewes with melatonin reduced the brain damage and improved udder-seeking behavior in hypoxic lambs, and other studies have shown an increase in brown adipose tissue and birth weight in lambs, especially when nutrition and photoperiod were suboptimal. So this brings me to talk about what we've done and the results. So melatonin stage 1, we had 3 treatments for the pregnant merino ewes, a control obviously with no melatonin supplement, a melatonin fed, where we fed the ewes 2 milligrams of melatonin daily from day 80 of gestation until birth, and then we had a melatonin implant treatment where they received a Regulin implant, which is 18 milligrams of melatonin at day 80 and at day 125 of pregnancy. And we did this across 2 lambing seasons, so autumn and spring. And I'll just give you a summary of the results. So what we found was, maybe a little bit surprisingly, was that lamb birth weight was not affected by treatment. The lamb birth weights for twins were quite high anyway, so we saw no significant improvements. But what we did see was that overall melatonin did improve survival to weaning. So survival to weaning with twins and the controls was around 73%, it was 73%. In the melatonin fed and the melatonin implant animals, it was 86%. Interestingly, when we looked at the data a bit more closely, we found that survival was generally similar for firstborn twins, but the melatonin was improving the survival of the second-born twin. And you can see this from the graph on the right-hand side. So purple is melatonin implant, green is melatonin fed, and blue is control use. And on the y-axis is lamb survival to weaning. And you can see here that survival of the firstborn lambs is fairly similar. If you look at the second-born, survival is very low for the control, it's below 60%, compared to above 80% and just below 80% for the male fed and the male implant, respectively. And then when we looked at the data a little bit more closely, I say we, I mean Tom, our PhD student, did, and we found that beneficial effects of melatonin on lamb survival seem to be most profound for extended labours, so anything over 90 minutes. And we found that extended labours increased the mortality of second-born twins in untreated ewes, but melatonin increased the survival of second-born twins, which experienced an extended birth process. And you can see this again from this graph here on the right-hand side. So the same colours, blue, control, green, melatonin fed, purple, melatonin implant, survival to weaning on the y-axis, firstborn for a short parturition and firstborn for a long parturition, no significant differences. Secondborn for the short parturition, there is a drop in survival, but it's not significant from second to first. If you focus on where the green box is on the second-born lambs that experienced a long parturition, you can see survival is much lower in the controls and significantly higher in the male implant and in between in the male, significantly higher in the male fed and numerically but not significantly higher in the melatonin implants. So that leads us to kind of support the hypothesis, go with the hypothesis that it is having some beneficial effects, in particular protecting lambs from hypoxia. So this, we got approval to go into stage 2. This trial was done at the Minipa Research Centre. We broke it down. We decided not to feed melatonin this time. We decided to use implants. So we had control animals with no melatonin implant. One treatment had a single melatonin implant on approximately day 90 post-joining, and the other treatment had 2 melatonin implants on approximately day 90 post-joining with no repeat treatment. And these figures just show you, pictures just show you the melatonin implant. So there's the implant, you can see how small it is. This is the applicator gun, and it goes under the skin above the ear, so behind the ear. And so I seem to have left the pictures out, but basically these animals were managed under standard husbandry. They were managed outside in fairly barren paddocks, and they were allowed to lamb naturally. We didn't impose any interventions, and we just went round twice a day and recorded lambs born and lamb survival. And we got a birth weight as well, about 4 to 15 hours after they were born. And what again we can see is that melatonin treatment did not increase lamb birth weight, so it's consistent with the previous trial. But also consistent with the previous trial, we did see an improvement in lamb survival. So you can see from the control animals that the number of animals born alive was around 94%, just below. Animals alive at weaning was around 80%. If you look at both implant treatments, almost 100% of animals survived the birth process. And in terms of weaning rates, 94% of lambs from the single implant treatment survived to weaning, and 93% of lambs from the second implant treatment survived to weaning. So what we've if you want to summarize it down, is that we found that implanting twin-bearing ewes with melatonin, including Regulin, on about day 90 of pregnancy, we think it does protect the lamb from damage caused by birth hypoxia. It increases lamb survival to weaning consistently in both studies by about 13 to 14%, which results in about an extra 26 to 28 lambs weaned per 100 twin-bearing ewes. We've seen this improvement in lamb survival both in intensively housed animals and also under field conditions. Regulin is commercially available, as a lot of you would know. It's fairly easily implanted. It's quite a quick process, it's just behind the ear. It's just what you need to keep in mind that its use in pregnancy is currently an off-label use. So I did do a quick return on investment. Again, these are back of the envelope, slightly more advanced than back of the envelope, but fairly close. So I've done 2 models. The first one is for animal producers that are already scanning for litter size, estimating about a mob of 1,000 ewes. We're estimating a 30% twinning rate. The cost for twin bearing ewes for the 30 to 300 is around $2,400 for the implant, plus maybe $1 in labour, and it's estimating a benefit of 84 additional lambs, so the 14% increase in survival. So in terms of profit, I've done it for Merino. Did the maths for Merino, terminal Merino, and maternal, and I did it for two prices per kilo of carcass weight. So obviously the bottom end of the scale for Merino, where it's a $5 per kilo carcass, the profit's around $2,000. At the opposite end of the scale, you're close to $8,000 profit at an $8 per kilo carcass weight. What I've also done is the break-even point, where you actually have to get what the minimum lamb survival increase is to achieve, at least get your return on your investment. For $5 per kilo, it's around 8%. For Merinos, at $8 per kilo, it's around 4%. For maternal, it's around close to 6%, and $8 per kilo, around 3%. So you need to achieve for a maternal a 3% increase in lamb survival to recover your costs from using melatonin when prices are high. When they're low, around a 6% for maternal and around an 8% for merino. The second model was based on people who are not scanning, who only scan for wet and dry at this point. And now it's obviously the cost goes up a bit to $2,900, because it includes a cost for actually scanning for litter size as well, plus the implants, etc. And it assumes the same level of increase. So again, it shows basically a similar pattern. If you have to now scan on top of what you're doing already, plus implant for a merino at $5 per kilo carcass weight for your lambs, you might make $1,500. For an $8 per kilo, you might make a profit of close to $500. For maternal, it's close to $300 at $5 per kilo. For a profit of $8 per kilo, for a price of $8 per kilo, your profit's around $7,390. And again, your break-even points increase slightly because you've now got that added cost of extra scanning. So I guess the question now is where to from here? Well, I'm just going to kind of go back sideways a little bit. So while we were doing this research and a little bit before and just after, other people have done some work as well across a number of countries and different species, different breeds, not species. So in merino, work conducted in Australia, in conjunction with SIVA and led by Alfonso Abacia, showed that if you give Regulin on about day 84 after the ram's introduced, so these are merinos in New South Wales, so an average of about day 42 to 84 post-conception, lamb survival over 2 years increased from 76 to 82%, and lamb growth to weaning was also increased. Work in Spain by the same group using Raza aragonesa sheep, which I think are a local sheep breed, shows that Regulin on day 120 post-mating increases IgG content in the colostrum, and there was a numerical increase in survival. The trial wasn't powered for survival, but when you look at it, the survival is higher. If you implant ewes 24 hours after lambing, you increase lamb weight at weaning by about 1.5 kilos. And if you implant the lambs directly, you do not have effect on their growth. So it kind of infers that the implant to the ewe is improving in milk composition or quantity. Another group in Greece, again using a local sheep breed, have shown that if you give give regular implants to ewes on day 0 being cedar insertion, and again on day 40, day 80, and at day 140 after that, increased fertility of heat stress ewes. So pregnancy rate was substantially higher, it was 77% versus 56% for the implanted ewes. Pregnant ewes gave birth to more lambs, so 1.65 versus 1.31 lambs per ewe. So lambs born as a percentage of ewes joined was higher, it was around 126% versus 74%. And that's most of the sheep work that has been done. There's a little bit of work has been done with fetal lambs, showing that melatonin does increase brown adipose tissue. And work in cattle has shown that higher doses of melatonin, so around 5 milligrams per day, rescue fetal growth of feed-restricted cows during summer. So that work kind of does support what we've done already. And so now where are we going from here? Well, we're going on to stage 3, and we're also working with an LA donor company project as well to commercially validate the benefits. So we're going to aim to quantify the benefits of melatonin supplementation during pregnancy within commercial farms, covering a range of systems, spring and autumn lambing, a range of breeds, merino, maternal, terminal, and a range of sheep production zones, so the high rainfall, pastoral and wheat sheep zones. Methods that we adopt will vary slightly across farms. Ewes will be scanned for litter size and fetal age. Implantation time will vary to suit the production system. We don't want to impose any additional movement of sheep or handling of sheep if we can avoid it. So we're aiming for around day 60 to 90 of pregnancy, maybe a little bit earlier if necessary. And data collection will also vary according to the production system. So the core data obviously will be lambs marked or weaned per implanted or control ewe, and any additional data based on opportunity and system will include things like ewe body condition score and weight at key stages, food on offer, the number of lambs born per implanted or control ewe to account for any scanning, variation due to scanning error or inaccuracies, and where possible, ewe and lamb weight at weaning as well. What we'll also be doing at the same time is we're doing a little bit more work with Regulin. We have a couple of projects funded by the Sheep Industry Fund. We're going to investigate the ability of Regulin implants when given to the ewe and the ram on their fertility and their thermoregulation during periods of heat stress and feed restriction. And we'll do that this summer through the summer of 2023. And we're going to do a similar model to what we've done already, which is combining fundamental research studies here at Roseworthy campus at the University of Adelaide and Turretfield Research Centre and other field study sites to understand the biological mechanisms. And then we're going to also do on-farm trials to quantify the benefits. So just to summarise, regular implants have the potential to improve twin lamb survival. They appear to be a cost-effective strategy. They may provide benefit above and beyond pest management, or the indications are that they may be beneficial when the physiology of the animal is under pressure, so limited feed availability, nutritional stress, heat stress, etc. We're going to validate this potential across a range of systems and sheep producing areas. And if the outcomes continue to be positive, CEBA will probably be required to apply for a label change to include pregnant ewes. So that brings me to the end. But just before I finish again, I would like to acknowledge MLA, Sheep Industry Fund, Davies Livestock Research Centre and SARDI, PIRSA, for funding these projects. The students involved, so Tom in particular, it's his PhD. This is Tom here with a beanie, loves holding sheep. And the other staff involved, postdocs, Elise Swinmore and Jamie Daly, Megan Sharkey, Emma Greenwood, Alice Weaver, and the staff and the academics, Jane Kelly, Simon Walker, Karen Kine, Kathy Gatford, and David Kleeman. So these are Simon Walker and David Kleeman up the top right. So thank you for your attention, and any questions?
Well, thank you, Will. That was a very comprehensive presentation that covered a lot of key points. And I just need to sort of pick up and just stress that, as Will said, that using melatonin during pregnancy is an off-label use. So we're not advocating people go and use it. We're advocating people wait and see what the research says, and then once, as Will said, once the commercial trials are finished and if we're proven to have benefits, then they'll have to apply for that label use, and it will become another tool for us to use. So just don't remember everyone to type your question in the chat box, and I'll ask that to Will. But I guess first of all, can you speculate on the mechanism that's causing the increased survival, Will? There's no change in birth weight. So what do you think is that physiological impact that melatonin's having that's giving you that improved survival?
Well, there's two potential mechanisms. One is around an improvement in brown adipose tissue levels. So melatonin reduces the breakdown of that during the gestation period. But because we've seen, and that would improve thermoregulation, but we've seen no difference in thermoregulation in the lambs. And given that we see the improvement in second-born lambs primarily, and also those with a difficult parturition, we are working on the premise that it's around the hypoxia impact. So melatonin prevents the programmed cell death that occurs as a result of oxygen deprivation. So prevents basically lambs that fail to stand up, because their brains aren't coping properly. But also, I think, stops that secondary death that occurs about 3 to 7 days after a hypoxic event as well. So I think it's the brain. It's protecting the brain basically.
Okay. And
it's not stopping the hypoxia. It just helps the fetal brain cope with it better. Okay,
right. So it doesn't stop it. It just gives almost like some kind of protective mechanism.
Yes, that's right.
Okay. So we have a question from Graham at Agravet. He's asking what other antioxidants could be used to improve survival with possible improvements. And do you know of any other likely candidates that that we could use?
I don't know about in terms of antioxidants, but the most likely compound at the moment is creatine. Creatine works really well in pigs, and it works well in rodent models. The problem with creatine that we have at the moment is it doesn't cross the placenta. So some of the work we're doing around amino acids is to try and increase creatine levels in the fetus as well. So that's why we're focusing on things like arginine, guanidinoacetic acid. So creatine levels, again, they also stop that programmed cell death. So they provide that occurs during oxygen deprivation. In terms of antioxidants, I'm not sure, but creatine is probably one of the better candidates at the moment.
Okay. And another question in relation to the trials is the question was, what was the lambing time for the trials the breed used, and were there any feed restrictions at the time of lambing? So was food restricted?
No, we fed them, we managed them optimally at Turretfield. They land in April, May, and August, September in Turretfield. So Turretfield is just outside Adelaide, near the Barossa Valley. And at Minnepa, they land in April, May. I don't think it was a particularly bad year, and the ewes were in fairly good condition. So they were supplemental fed as well. So it probably was best management practice at both sites.
Yeah. And I think also if people wanted to chase up on the papers that Tom and your group wrote in Journal of Animal Science, they were listed in the February edition of the Sheep Reproduction RDNA alert, which you can get off the SRSP website. So another follow-up question. So is the melatonin rate responsive? So they're wondering if could we use 9 milligrams rather than 18 milligrams to lower the dose and get the same response, given that lowering the dose would be more cost effective potentially?
Okay, so the 18 milligrams is just an implant, it's an elastic implant, and it releases the melatonin slowly. So we've only looked at the single dose and a double dose. We haven't looked at a lower dose. And all that really does is basically ensures that there's a constant level of some release of melatonin during the day at a fairly low level. And all it does is supplement the nighttime levels a bit. So we haven't looked at lower doses. But we did, when we fed it 2 milligrams a day as well.
Okay. And you mentioned the commercial sites. When do you have some of those started?
Not well. Some of them have. I don't know when the data will be in. We're still trying to work. A lot of them. We've worked with a couple of ram breeders, so they're dealing with the ram sales and stuff. So we haven't got full data yet. But most of the farms will start. Animals will be joined. I think the earliest is November, and then some will be joining in February. So it ranges between November and February this year. Well, this year and next year.
Okay. And just the focus of the work so far has been on twin lamb survival. Do you think it would have a similar sort of benefit for triplets, which are sort of, I guess, a higher proportion of births if we go into some of the maternal breeds?
Yeah, so we haven't looked at triplets, but I suspect the impact is probably higher for the birth process on at least the third born triplet. That would be a twin. So I think the potential is definitely there. I forgot to mention we have done this work. The work at Minnepa involved singles as well, and we saw no benefits at all. So that kind of pushes it that way. But so I think yes. Short answer, yes. I think it probably would work for triplets. We saw no benefit in singles. But the Spanish group who did the work in New South Wales, I think they implanted singles and twins, so their improvements were across the board.
Yeah. And I'm glad you gave me that little segue to the Spanish team, because Alfonso, as everyone can see, has just sent that message into the chat, and they've congratulating you on results for your trial, Will, but also they've just finished a new experiment implanting 1 month before lambing, and they've seen that sort of higher IgG content in the colostrum of the implanted ewes. So it looks as though it's a good story. But as I said before, at the moment in Australia, this is an off-label use, and so we need to wait and see what the trials say, and then wait for CEVA to get that use extended for the product. So do we have any more questions from anyone? Okay, so that's the case. I'd like to thank everyone for listening in to the webinar today. This was our first one in the series. So our goal is to run a quarterly series. So our next webinar is scheduled for the 19th of November, and the focus of that will be on breeding new fitness and new mortality, and we'll have a couple of presenters, Andrew Whale and Mary McQuillan from Livestock Logic. So that's something to put in your diaries on the 16th of November, and I'll be setting up the details of that, and we'll be putting it up on the MLA website as well. And as I said, this recording today the webinar has been recorded, and I'll make sure I can get a recording up on the SRSP website as well. So thanks again, everyone. And again, if you have any topics or something sheep reproduction related you would like more information or want a potential webinar on that topic, please don't hesitate to get in contact with me, and I'll do my best to make it happen. So again, thank you, everyone. Thank you, Will, for your very informative presentation, and I hope everyone has a great day. Goodbye.